Speaker is Dr. Sophie Molholm. She is a cognitive neuroscientist at Albert Einstein College of Medicine in the departments of pediatrics, neuroscience, and psychiatry and behavioral sciences, and director of the NIH-supported Rose F. Kennedy Intellectual and Developmental Research Center. Her innovative investigations into the mechanisms underlying perception and cognition in many disorders, including cystinosis, have been instrumental in designing therapeutic interventions. Dr. Molholm.
Thank you, Rick, and thank you for having me here. Let's see, let me just figure out this. Got it, okay. Okay, so I've been coming to these cystinosis meetings for a while. This is a picture from the meeting in Philadelphia just a few years back with my collaborator, Dr. John Fox, who's here today, Ana Francisco, who's done a lot of the work that I'll talk about, and then this is Dennis Cregan, who's also been working on some of our projects on cystinosis. And I just want to say thank you to Rick for bringing me and my collaborators into the world of cystinosis. It's been a wonderful journey. The families are wonderful to work with. The other researchers are amazing. And the CRN and Christy Greeley for all the support that we've gotten for our work. So you all know a lot about cystinosis and how the accumulation of these molecules leads to dysfunction of different organs. And of course, this has been most dramatic for the kidney. But through all of the innovative work that's gone on in the past 20 years or so, remarkable interventions are now available, and the lifespan of individuals with cystinosis is much improved. So, cysteamine accumulation also impacts the brain and neural function, but to much lesser extent. But now that there's prolonged life, we can turn our attentions to this problem, which, while not as severe, is certainly impactful and one that we want to understand and hopefully see how different interventions also improve brain function to the extent that it is impacted. So how do we know that brain function is impacted? In part, we are aware of this because of the different neurological complications that are associated with a diagnosis of cystinosis. And I can highlight a few here. Things like cognitive functions, including visuospatial, visual memory, and visual motor problems. Academic function is also somewhat impacted, particularly arithmetic and spelling. And there's some evidence of memory impairment as well. And we've also found, and there are also reports of some concerns around executive functions. So significant difficulties are common in these areas. And this, this is just repeating what I said, but that really also can extend into adulthood. And here's some evidence from other studies just showing how in individuals with cystinosis, you can have this discrepancy between verbal comprehension, so verbal abilities, and some more performance and visual related abilities. And when you have these kind of discrepancies, that can be particularly frustrating. But again, you can see that some of the deficits are in working memory, performance, and processing speed. This is just more evidence that there's a discrepancy between verbal ability and performance and nonverbal abilities. So what are the behavioral neural correlates of some of these problems? There's evidence for subcortical and cortical atrophy. Chiari malformation, white matter abnormalities, and also some evidence of atypical electrophysiological activity. A lot of this early work, or original work, was done by Doris Trauner. So I need to acknowledge all the work that she did that really set the stage for looking at brain function in this group. So we came to this problem space with our tools of electrophysiology, which is looking at brain function by measuring electrical activity non-invasively. And we really sought to understand basic sensory processing. So can we observe any deficits, or when do we see differences in brain function in these groups? Is it during early sensory processing, or is it not until later cognitive processing? So these are just symbolizing auditory and visual processing, which I'll be telling you a little bit about. And then executive functioning, which I probably won't get around to telling you about today. Here's the methods we use. So you can see we have a child here, one of our participants, a couple research, a research coordinator, and Anna, a postdoctoral fellow at the time, and we're going to record brain activity. Am I talking too close to the mic? Okay. So what do? Here's the ongoing EEG. We present stimuli, and then we average the brain response time-locked to those stimulus presentations, and we get something that looks like this. I'm going to be showing you a lot of these squiggles, so let me just unpackage it this first time. On the x-axis, we have time in milliseconds, and on the y-axis, we have the size of the signal, and then we have whether it's negative going or positive going, and then I'll refer to these as kind of components. So P1, M1, P2, they're commonly named by when they come in time. So that's the first positivity, the second positivity. And then I'll also be showing you some brain maps like this. So we record from many channels. This is the response from one of those channels, but I can also show you a topography of the distribution of the electrical activity across the scalp, which gives us an idea of the intracranial generators of the signal. And first, I'm going to start by telling you about some auditory studies that we've done, looking at auditory sensory processing and auditory memory. And so for this, we present tones, and what we're looking at is how does the brain process these tones automatically. So generally, the person can be watching a movie or something. We just want to see how's the brain working to automatically process this information. And we can look at, so to do that, we look at what we call the N1, and then we can also look at sensory memory. So when you have a series of tones and you change the tone, the brain responds to that change, and it responds to that change based on the memory that it has developed for the preceding tones. And that we call the mismatch negativity. And here's just a little schematic of the paradigm. So you have a tone that's of one duration, and then you have subsequent tones. These are standards repeating of the same duration. This is a deviant. It's longer in duration. And then we can present these at different rates. So this is at a rapid rate. When we present the stimuli at slower rates, in order to detect that this is a duration deviant, memory has to be stronger. So in this way, when we present them at a slower rate, we're taxing memory, and we can see, are there differences in memory performance? This is just a little bit about our group. I'm not going to go into it too much, but I do want to mention that here we are looking at brain function in a rare condition, and therefore our sample sizes it takes a lot of work to bring participants in, because there aren't that many people, and they're across the nation or across the world. So we've been flying people into the lab, or we go to family meetings to collect data. But because it's a rare disease, our age ranges are large, which is not what you want, ideally, but so we'll be looking at data from childhood to early adulthood. And what you can see here is the mean age, and that our samples are about 25 for our controls and 19 for the cystinosis group, which is a nice robust number for this kind of study. You can also notice that IQ is a little bit higher in our neurotypical group compared to our cystinosis group, and that's pretty standard across all the data sets that I'll be showing you. So here are some of those squiggles I was telling you about, and I'm just going to focus in on this first one. And so you can see the stippled line. These are the neurotypical responses, and these are the data from the cystinosis group. And the first thing to notice is that this very early response here is very similar between the two groups. What that tells us is that auditory processing is intact in the brain. At 100 milliseconds, the brain's already done a lot of processing of this input, and everything looks completely typical in our cystinosis group, and we see that replicate across our different stimulus presentation rates. And so this is just kind of highlighting for you the similarity. Then we can also look at the mismatch negativity, which I mentioned to you, and this is our index of memory, auditory memory, and how well is that working. And again, we're looking at different presentation rates as we go from rapid presentation rates to slow presentation rates. As we go slower, we're taxing memory more. You have to hold on to that information for a longer period of time. So here is, this is our index. It's called the mismatch negativity. We get that by subtracting the standard response from the deviant response. And you can see that in our control group, here are the mismatch negativities for those three conditions. And here they are for the cystinosis group. And you can see, just eyeballing it, that there's a difference there. And in particular, the auditory sensory memory for the two slower presentation rates is much diminished in the cystinosis group. This sample happens to be children with cystinosis. And we also did the same study. I'm not going to show you the data, I'll just describe it, in adults with cystinosis. And what we found there is that there's some memory function appears to improve from childhood to adulthood, so that we don't see as big a difference here. So that's some very hopeful, positive news. So just pointing that out to you here. Then another question we might ask is, if we see brain differences, do these correlate with any of our cognitive measures? And what I can tell you very briefly here is that when we look at verbal IQ, perceptual reasoning, there's no correlation. So, and that's whether we look at the mismatch negativity, that auditory sensory memory, or basic auditory sensory processing. So now we see auditory processing is very typical. There's some evidence of some deficits in auditory working sensory memory. What about visual processing? That was the next question that we turned our attention to. And actually, we kind of found evidence for visual processing differences in a task where we weren't focusing on that. So this is what we call a go, no go task, and individuals are getting presented images, and they're supposed to press a button to each image as it comes along and withhold their response when it repeats. And what we found here, here's a little bit about our sample. You can see it's pretty wide age range, 7 to 36 years of age. But what we found here is that the red trace is the visual evoked response. So the brain's response to the visual stimulus for the cystinosis group, and the purple trace for the control group. And so what you can see is there's a really marked difference here. And that difference goes in the positive direction where we have a significantly larger response to the visual stimulus. So this really captured our attention, as you can imagine. Here are some of those topography maps I was telling you about, and you can see, it's not just an artifact of the channel that I'm showing you. The scalp distributions, that yellow is where you have the biggest signal. They're much larger in the cystinosis group, the magnitude of the response compared to the control group. Here again, we asked, does this correlate with cognitive measures? And we found that very clearly there's a correlation with the amplitude of the visual response and age. That's known from the literature. It doesn't matter which group you're looking at. And then we also found that it did not correlate with either verbal IQ or nonverbal IQ. So a little bit surprising because we see that large difference. You might expect to see it correlate with a cognitive measure. So why might it not correlate with a cognitive measure? Maybe we're measuring the wrong part of the visual response. Maybe the cognitive measure we're using isn't the most sensitive. Maybe there is no relationship. Maybe our samples aren't large enough, et cetera. So these things can just be a sensitivity issue that you haven't measured exactly the right thing. So you wouldn't want to conclude definitively that this means there's no relationship between this altered brain function and what's going on behaviorally and cognitively. But to this point, we don't have evidence for that yet. So we wanted to explore a second data set and see, do we replicate this finding in a whole set using a completely different visual paradigm? So here we just present checkerboards. And they're just coming on for very brief periods. And we presented them at different rates of presentation. So we could look at something called visual adaptation, which I won't go into too much here. Here's a little bit about our sample for those of you who are interested. Again, you can see that the IQ scores are a little bit lower for our cystinosis group compared to our control group, but they're in normal, pretty good range. And here are the data. There's a lot of it here, but let me just draw your attention to the top panel. And that's just looking at the visual evoked response over several different channels in the control group. The different traces that you see are for the different presentation rates. Again, I'm not going to focus on that. So just kind of give yourself a global bird's eye view of the response, averaging across all those individual waveforms. And you can see that the control group response is much smaller than the cystinosis group response. So showing that same thing that I showed you for the other data set. So pretty striking difference between groups that we see here. I just did that to enhance where you can kind of see the difference best. And again, if we look at scalp topography maps in the cystinosis group, you can see that yellow is much brighter for the cystinosis group than for the control group. This is just showing topography maps over time. Why do we look over time? Because the brain is processing information very rapidly, so it may change over time. But you can see that the differences are pretty consistent between 100 and 250 milliseconds. You've seen that. And then this is just showing if we take each of those different traces for the early portion or the late portion, we kind of divided it up here. Here's the early portion. Here are the responses, the amplitude of the response for the cystinosis group compared to the control group. So just really bringing that home. It's more in the early portion of the brain response compared to the late portion. So what can we say about this finding? Well, visual perceptual indices are often significantly lower than verbal indices, as I said previously, in individuals with cystinosis. This pattern, this kind of clinical pattern, emerges early in development and persists throughout the lifespan and regardless of age at treatment onset, so far from what we can see in the data. There are significant difficulties described in domains related to visuomotor, visual spatial, and visual memory skills. So what does this signal represent? Maybe it represents visual cortex hyperexcitability. Really, we need to do more work, kind of some follow-up studies to really unpackage what's going on here. But just to point out that visual cortex hyperexcitability has been associated with photophobia in individuals with migraines who, in some studies, have been shown to similarly exhibit these increased visual evoked responses. And as we know in cystinosis, this is a significant problem, this light sensitivity. So what are some future work that we might do and implications here? We need future studies to investigate visual processing pathways in more depth. So maybe see, is it a dorsal visual function or a ventral visual function? Are there certain types of stimuli that are more challenging than others? So we can employ stimuli that will allow us to dissociate these different visual pathways. But we also need to keep in mind that any visual studies that we do should take into consideration this sensitivity to light. And indeed, when we had participants in the lab, they would sometimes complain about the brightness of the stimuli. And we didn't know in the beginning that this was something we needed to think about. Now, obviously we do, and we will certainly, adjust contrast accordingly for future studies. There are also implications of being aware, we know behaviorally and clinically, but now we also know based on brain responses that we need to make the classroom and professional settings appropriate for people with cystinosis, or be sure to apply tools to allow people to perform most effectively. And then based on this really large signal that we found, these facilitate some work that's going on at the University of Rochester in John's lab there, which we can refer to as back translational work in mouse models. So I'm going to just very briefly present some preliminary beginning work that has been done. This involves John, of course, and Landy and Haley. Sorry, I blank on names. Landa and Haley. So Haley's a medical PhD student, and Landa's a senior researcher in John's lab. And so what's the point of this? So if we can compare evoked neural responses in humans and mouse models of disease, that may help us elucidate the neurogenetic underpinnings of neurological disorders. Shared endophenotypic expression provides a measure of validity of the animal model. And having an animal model is really critical, obviously, to being able to do more invasive therapeutic approaches, and also things like high throughput testing of pharmacological interventions with using this validated neuromarker. So do those interventions not only improve kidney function, for example, but do we see it impact the visual evoked response and normalize it? This is just showing you the setup. This is this little electrode that gets implanted on the mouse model, and then you can record brain responses to different types of stimuli, and this is just showing some of those brain recordings. This is the current setup where visual stimuli are presented on a monitor. There's the mouse. And Haley could tell you a little bit, and Landa could tell you a little bit more about this if you want to seek them out later and hear more about the work. And the really cool thing is that then you get visual evoked responses from the mice. This is coming from this channel, which they say represents activity in B1, so I believe them. and right now they're using different contrast levels. And what I notice here is that the response looks most like the kind of visual evoked response that we see in humans at this 25% contrast level. And so maybe it was suggested that the response might be a little bit saturated as we go to higher contrast levels. But anyhow, when you look at the wild type versus the knockout mice, okay, very, very preliminary data, but you can see that it does appear like there's a larger response in the knockout mouse. So I don't want to take that too far, but that's especially because I can't tell you that much about the data, but that's really exciting work, actually. And then I wanted to click on this hyperlink to bring me toward the end of the presentation. Is there anybody here who can click on it? Because I don't have a keyboard to do that. In the back, maybe?
Which one you want to click on?
Yeah.
If you just click. Thank you. Perfect. So I've just briefly told you a little bit about some of this work we've done looking at sensory processing, where we find these pretty major differences, but just in one sensory modality and the one you might expect, indeed. But I wanted to end on a very kind of hopeful note. So in the lab, we've had this great experience of working with families that come. And a lot of the families, as we had new studies going on, came back for a second visit. And that was over the course of 4 years. So that gave us the great opportunity to compare measures that we had made at time 1 and time 2 and see, is there any evidence of kind of downward progression, and what we found. Okay, so let me just show you this. Anna's here in all of these. She's actually left science, very sadly, but she's still involved in helping us with some of the cystinosis work. And it certainly wasn't because of working with cystinosis that she left. She loved the families and the foundations. And here are these kids, at 2 different ages. And just looking at verbal IQ and nonverbal IQ, which was kind of the most basic thing we could compare, what you see is that there's no decrease. These are very stable over time. So really, I think, suggestive of the efficacy of the treatments that have been developed, and great news. And this is just, I think, Bill or someone might have shown a slide similar to this, but if you look at total IQ in individuals with cystinosis and age of cysteamine start, you can see that IQ is in the normal range when you have very early onset of treatment. So another kind of plug for early detection. Okay, so to conclude, there are subtle differences in brain function that are likely contributing to cognitive deficits. I didn't tell you about the work we've done in executive function. We've done quite a bit. And there we find subtle differences, nothing really profound. Things like how quickly recognition of making an error occurs. So it's little differences like that. There are prominent differences in visual sensory processing and sensory memory. Oops, that's just my timer. And slower processing of errors, as I was just mentioning. So based on these data, recommendations to children and adults with cystinosis might be, in most respects, your brain is working very well, but give yourself more time to complete tasks and use organizational tools to help keep track of elements of more complex tasks. And then I wanted to end by saying we are very grateful for all the support that we've gotten from the CRN and to all the participating families for coming to the lab and making the time to participate and just being, so fantastic. So. Yes.
You want to put up the next slide?
Yeah, you spend time, so most of us are not as familiar with your field as some of the other fields of some of the other discoveries. But you spend time addressing potential correlations between the evoked potential differences you get as compared to gross cognitive skills, which I would imagine is difficult to start with. But in terms of the dissimilarities or similarities that you see here with cystinosis, does this track with other diseases? In general, do you find better correlations with the electrical differences and grossly measured cognitive
That's
differences?
a great question. Thank you for asking that. No. It's quite challenging to find correlations between basic sensory processing and cognitive measures, and so we do find them sometimes, but it's not surprising if we don't find them, really for the reasons I mentioned. Are you measuring, is your, if you want to call it outcome measure, a good measure of that little brain signal that you have? And there are so many things that contribute to the behavior that you're measuring, and so that brain signal might contribute a little bit, but if you have an N of 30, are you really going to be able to see that? So it's a challenge for us to be able to recognize those relationships. So when you have a huge relationship, basically the signal has to be really big in order for that to emerge, and sometimes we're lucky and it does, and other times not so. So that's why one needs to be cautious in drawing too strong a conclusion and saying, I would not conclude that there's no relationship here. It's just we haven't found the right measures, or it's not sensitive enough when we don't show that relationship. I think there's one more question. Do we have time?
Quick question. Do you think the degree of eye disease correlates at all with the visual processing differences, or?
That's something that we need to look into, and we really want to understand better. I think that's a really important question, but I don't have an answer just yet. Okay, thank you.